Hello, in this video we will discuss about the cell death signaling pathway and growth pathway. So the cell proliferation and growth. So how it's possible? Let's begin to understand and unwind the major concept for understanding the apoptosis. So the cell death is natural process of old cells dying and being replaced by new ones after the cell division, and the damaged cell will be recycled and or may result from such factors such as disease, injury, and death of organism, which that is very important. And this all thing is related to the cell death. So this cell death signaling pathway is the catabolism. In this way, the type of anabolism is known as the catabolism, and cell death is known as the apoptosis. So the cell death can be two types: autophagy or apoptosis. So the autophagy means the cell will destroy itself, while the apoptosis means the cell programmed cell death. So let's begin to understand another major concept here is the cell growth means proliferation and differentiation, and the progression during mitosis and meiosis. So the cancer cell relation with the proliferation and the sudden increase of the growth factor will lead to cause cancer. We will also understand. So the cell death is contain autophagy or apoptosis. So first of all, we should need to understand about the apoptosis. So the autophagy is the itself cell will be damage itself for because damage organelle etc. But here the apoptosis contain programmed and necrotic cell death. So the programmed cell death is after damage DNA, organelle, and other will programmed to die of cell. So the necrotic is the non-physiological process that occurs as a result of infection or injury. So let's begin to understand. Here is I am drawing the bigger picture of the cross section of the cell for understanding good. So let's begin to understand with the plasma membrane with several receptors for giving the function separately. So first of all, here is the different type of receptor we will understand one by one. So number one, here is the plasma membrane extracellular, and here is the intracellular cytoplasm, and inside is the nucleus contain nuclear membrane. Its membrane contain bilayer of phospholipid which that is embedded the receptor for giving the signal, because without receptor it is impossible for giving the signal to inside of the cell for changing the cell physiology. So here is the recruit others protein through insulin like growth factor 1 when bind as a ligand with the receptor tyrosine kinase will give the phosphorylation and docking the other protein phosphoinositide 3 kinase to phosphorylase the AKT and AKKA. And in this way the protein kinase C will be activated also to activate the necrotic factor kappa B and the IKB. So the IKB is also stimulated, and it will be bound with the DNA for transcription of the necrotic factor kappa B. So it means that is the protein kinase C is basically used for necrotic factor kappa B for proliferation of cell. So let's begin to understand the death of the cell. This was the cell growth process. While here is the interleukin binding with the antigen presenting cell, for example, which that is, and interleukin is basically cytokines bind, and JAK and STAT pathway will be occurred. So the JAK will be phosphorylase after the JAK docking the STAT3, and in this way the STAT3 will be recruit and separate, and in this way the dimerization will be occur, and it will be bound with the transcriptional factor as a transcriptional factor with the DNA with the specific genes for the promoter region to promote the gene transcription for translation. So this STAT3 is the type of dimers So the dimerization. So this is the type of STAT3, which that will be activate the P53. So this is we are going to understand the cell death. So P53 is the important protein which that will be stimulate the other DNA after the phosphorylation to other DNA segment gene bags. So the bags gene will be activated by this. We will discuss later here. So here is the another is the death factor receptor is the TNF bind with the fast receptor. So this fast receptor will bind to increase the fat protein, and this fat FADD is the protein which that will be stimulate through gene expression. And after this here is the stat3 and 1, stat3 and 5 is involved for this all mechanism also. So here is the fat will stimulate the caspase-8 protein. This caspase-8 protein phosphorylation to trigger other phosphorylation of protein. And here is the stat3 and 5 is basic type which that stimulate the BCL-XL. And in this way the mitochondria which that is bind with this and move inside and to release the cytochrome C through binding the bags. So the bags will bind and bags will stimulate from the P53 gene activation to produce bags, and BAD is also produced from that by the stimulation of P53 protein. And this P53 protein will bags will be bound with the mitochondria will release, and also the abnormally sensitivity will increase the beam, and this beam will also trigger the mitochondria for increasing the cytochrome C. And after the increasing of the cytochrome C will be the Caspace9 will use for the apoptosis, and the BCL2 is used for the inhibition. So all these things is related to the death of the cell. So here is the several gene are present: P21, cyclin, and CDK2, P27, and P15, and P16, and cyclin D and cyclin CDK4. So let's begin to understand with the G coupled protein receptor, which that is also important, which that bind the chemokines, hormones, or neurotransmitters. This all things is the ligand will be bind, and the trimeric subunit trimeric protein will contain alpha subunit GDP will be activated to GTP will bind with the adenylate cyclase enzyme to trigger the cyclic AMP. After the triggering of the cyclic AMP will stimulate the protein kinase A. And protein kinase A basically is trigger the CRAB, and this CRAB genes will activation to cell proliferation. While on the other hand here is the growth factor receptor. Here is the receptor tyrosine kinase which that is after binding the growth factor, tumor growth factor alpha and EGF and serotonin. After binding the recruitment of the growth receptor binding protein 2 and to recruit the SOS and RAS. After the RAS will convert, RAS will stimulate the RAF by phosphorylation to stimulate the MEK, and this MEK will stimulate three type of protein: MEK kinase, MAP kinase, and MEK kinase, which MK kinase, which that stimulate the different gene, for example NFKB, CRAP, and mice, ARC, FOS, JANX, and beta catenin, TCF. JUNX, which that is also all things will need to cell proliferation, and JANX will stimulate the JUN. So here is the integrin will bind. Here is the extracellular matrix, and this receptor is known as the extracellular matrix recognition receptor. Will stimulate the CDC4 2 and the FIN and SHC protein and FAC and SARC protein and RAS, as well as the Pi3k, which that is the phosphoinositide 3 kinase, which that will be RAS also stimulate the RAF. So remember about that. And on the other hand, the RAF will be stimulated to again the stimulation all of genes. While on the other hand, here is the free cell containing Vent receptor, Vent beta catenin pathway will be occur. In this way, here is the phosphorylation will be occur, and this phosphorylation will be glycogen synthase kinase enzyme 3 beta will stimulate the APC, and this is APC protein will vent beta catenin pathway will be occurring to produce a beta catenin protein. After the binding with a beta catenin protein with TCF to stimulate the beta catenin TCF, and this will lead to cell proliferation also. And here let's begin to understand the viral infection. So after this viral infection with a specific receptor we recognize through a spike protein of virus, for example. So the virus will move inside is known as the endocytosis. So in this way the endocytosis here is move inside. After the clearance by the phagocytosis as well as the other things will mechanism to kill the bacteria or pathogen, and after the killing to produce the antigen content, and this antigen representing cell will stimulate, and basically the piece of antigen will be a piece of antigen will be a show off. So here is a showing to the CD8 cell, which that is after the stimulation from the CD4 by the recognition of the antigen. After the antigen recognition to bind with the MSC class 2 molecule, 1 molecule after the MSC class 2 molecule. So this is the MSC class 2 molecule will stimulate the other Caspace activation, and this Caspace activation is used for the cell death, and it is stimulated by the here is a fast trail bind with a trail fast receptor, which that is basically the trail is used for the Caspace activation and Caspace activation, and fast molecule will stimulate the Caspace 8, while on the other hand the cytochrome C stimulation for the Caspace 9. So in this way here you can see this is the CD8 cell will produce the grain enzyme and perforin. Perforin bind with the cytoplasm, plasma membrane, to move inside the piece, basically to move inside, and the mitochondrial pathway will begin. This mitochondrial pathway will begin to binding with the bags after the transcription by the death-associated gene activation, and in this way here is a piece of antigen will be bound with the mitochondria and to mitochondrial pathway, and in this way the bags will bind here. So here is the Caspace 9, which that is Caspace 9. Here is the cytochrome C. The cytochrome C is basically trigger the after the efflux of the cytochrome C from the mitochondria. So here is the same mechanism. Here is the bags will stimulate by the P53 gene activation. After the activation to bags produce, and bags will bind with the mitochondria here. So here you can see the bags will bind to efflux of the cytochrome C protein, and the cytochrome C is basically stimulate the Caspace 9. So same mechanism which that is we had discussed before. So in this way here is the cytochrome C which that stimulate the Caspace 9, and after the Caspace 9 and Caspace 8 activation to kill the infected cell. So suppose this is the infected cell, for example. So the abnormally sensor is the beam is also used for this here you can see, and the beam is the protein is the abnormally sensory protein. And after this induction of apoptosis will be occur, and this all thing is related to the apoptosis, but also we had discussed about the cell death and cell growth, so proliferation also. Thanks for watching. Please make sure to subscribe, like and share, ask the question and give the feedback. Bye.